All right, well, good morning or afternoon, depending on where you are. We're excited that you are able to join us today for WQRF's third summer school webinar. Today's webinar will actually feature some WQRF research. Wanted to offer you an update on the PFAS surrogate study that we've been conducting. So special thanks to our sponsor, Calgon Carbon Corporation, for their sponsorship of the entire WQRF Summer School series. We're grateful for their support, and also to all of the WQRF contributors who are likely on this call today. We appreciate you and thank you for your support. So quickly, a little housekeeping before we get started. As you know, we're using Zoom today, and we'd love for you to include any questions that you might have for our speakers in the Zoom bar. There is a Q&A button. Please use that to enter any questions you have. We will answer all of those questions towards the end of the hour with our speakers today. Additionally, look to your email. Towards the end of the week, we'll send you a link to the recording. But as always, we do post all of our webinar recordings on our website at wqrf.org/webinars. So either way, look to your email or check our website towards the end of the week, and you'll be able to find the recording there. Today's webinar features the work of two really awesome human beings, but also great researchers, Dr. Majid Moshemi and Dr. Ehsan Esfahani, on our nearly completed WQRF study to identify PFAS surrogates. Instead of trying to develop individual reduction standards for each of the currently known PFAS, the objective of this research is to investigate development of surrogate compounds as a mixture that will cover multiple PFAS for product certification testing for activated carbon and anion exchange media. So really quickly, a brief introduction for our speakers. Dr. Moshemi is a professor at the University of British Columbia and is the scientific director of Community Circle, a nonprofit organization focusing on achieving socioeconomically and technologically sustainable outcomes in water health for Indigenous and rural communities. With world-leading expertise in the area of drinking water quality and treatment, he has pioneered the deployment and implementation of several water treatment projects. He is also the primary investigator of our PFAS surrogate study and has been overseeing the overall research and funding of the study. And Dr. Ehsan Esfahani is a postdoctoral fellow at the University of British Columbia. He has completed his PhD on novel treatment technologies for PFAS and is involved in several PFAS treatment projects based on adsorption and destruction techniques, as well as conducting field studies at PFAS contaminated sites. He was responsible for the various stages of the work, including the literature review and the overall day-to-day execution of the research, as well as overseeing the activities of the co-op student. So without further ado, I'm going to turn it over to our speakers. Thank you so much.
Thank you, Callie, for a nice introduction. I appreciate that. It's a great pleasure for us to be here and to present some of the work that we've been doing as part of the study that has been supported by WQRF. As Callie mentioned, we are both from the Department of Chemical and Biological Engineering at UBC in Vancouver. Just to reiterate, the purpose of this particular project that we've been involved is to determine PFAS surrogates that can be used as standards to evaluate the efficacy of adsorption-based processes. Now, I know many people on the call are somewhat or fully familiar with PFAS, but I'm just going to recap some of the basics around PFAS and why we are conducting this study before delving into the results of our research. So PFAS represents a group of chemicals. The latest count is over 14,000 compounds that have carbon fluorine in their structure. In fact, the formal definition is that if there is any molecule that has either methyl or methylene-based carbon in there, so CF2 or CF3 is categorized or is defined as PFAS. So one example of that, which you see here, is PFOA, which has two parts. Generally, all of these PFAS have two parts in their structure. One is the tailpipe part that we call, and those are more hydrophobic, and that's where the carbon-fluorine bond is. And the functional group part that is generally hydrophilic. And given the strong bond between carbon and fluorine, these molecules have very unique properties. And that's what they are so great, and they are used in many applications in our daily lives. They have great thermal stability. They are resistant to chemicals. They're both hydrophobic and lipophilic, and that's what makes them, again, very applicable to a lot of products and applications that we have. While they are hydrophobic, but they're at the same time water soluble, and that's where the problem comes, and also they are very recalcitrant in the environment. Their surfactant properties, of course, gives them unique features for fire retardant applications. Now, many consumer products, many industrial products are using PFAS in their structure. It's known over 200 categories of products with 1,400 or around 1,400 manufactured PFAS. And of course, as I mentioned, the number of PFAS in the environment are a lot more, but those are more transformation products or precursors that are there. So they're being used in various applications, whether they are as stain resistance or you name them, food packaging, grease repellents, and so on, and also, of course, firefighting foams. Now, their chemistries are very different. So when we talk about PFAS, we are not talking about one or two types of molecules. And they come in different chain lengths, basically. And so for as few as 2 carbons in their structures to many tens and mores. And they come with different end groups, whether carboxylic or sulfonated ones as well. Some of them are branched, and some of them have oxygen within their structure. So these are etheric groups. And the famous ones are, of course, are PFOA, PFOS, GenX, and the regulated ones that are currently regulated by the EPA. But again, these are examples. So these different chemistries give them different properties. And those properties are not only come when they are being applied, but also when we want to treat them in our water. So we'll talk about those a bit later on. So what are the concerns? I mean, certainly you have heard that there are many health concerns associated with PFAS when we are exposed to PFAS, and many of us have PFAS in our blood. In fact, estimated that over 98% of our people, even in northern areas, are basically have PFAS in their blood. But what the exposure routes? There are multiple exposure routes. Water is being one of them. So certainly we get them from food, from our indoor dusts, air, and so on. So variety of exposure routes. Water, as it's shown in this graph or in this table by a study that was done, is not the major exposure routes. However, the challenge is that if PFAS are in the water, they are literally all absorbed in our body. So that's the challenge comes. So that's why there are increasingly stringent regulations around PFAS and the applications and their presence in our water. The latest one and the most notable one is the one that EPA has introduced this past April and targeting 6 PFAS. Now, PFOA and PFOS are the ones that are called legacy PFAS, and these are the most stringent ones at 4 parts per trillion. And again, these are regulations for the removal, but if you look at the MCL goal, or maximum concentration limit goal that EPA specifies, it's around 0. So what it tells us is that or what studies have shown is that even small amounts that are in the water can be harmful. Now, there are various other ways as well, like hazard index that is shown in this slide, how it's calculated for 4 other PFAS as a mixture should be less than 1, in addition to 3 of them being 10 parts per trillion. So based on the regulations, as it was discussed in the previous webinar, the utilities are required to monitor for PFAS and notify the public of the levels that exist in their water, and of course, take actions to eliminate or reduce the levels of PFAS. Now, in terms of occurrence, there are more and more studies being done to determine what levels of PFAS are present in our water. And this is one map that is produced and it's live, literally, it's updated all the time, but environmental working group that shows different areas with different sources that have PFAS. And it's color-coded, shows that which ones are over the limit, which ones are not over the limit, and which ones are, for example, military sites. So with all these, what are the treatment options? Now, given that PFOA and PFOS, as I mentioned, these are legacy compounds, and there have been many evidences and studies that they are harmful, and in fact, they have been banned in terms of their production a number of years ago. So there are plenty of data available on the removal efficacies. Now, with the alternatives that have been manufactured, that are being used, and also being identified or detected in water, there are more data coming as well. But in general, based on EPA and other regulatory agencies, 3 technologies, either at the larger scale or a smaller scale, are considered viable and implementable. These are granular activated carbon-based processes or carbon-based processes that are relying on absorption of PFAS on solid media, anion exchange resins or anion exchange process, which can capture through absorption and exchange of PFAS with the counter ions that are on the resin. And of course, high pressure membranes, notably RO, reverse osmosis, being a compound that can, a process can remove PFAS. So these are already established technologies, and there are evidences that they can remove PFAS. However, when it comes to adsorption-based processes, whether activated carbon or anion exchange, there are factors that play a role in terms of efficacy of the removal. Now, one and most notable one is the PFAS chemistry or structure. So whether it's the chain length, so there are various chain lengths, and there are basically how many carbons they have, functional groups, what type of functional groups we are talking about, whether they are branched or linear structure, they have oxygen or etheric groups, or whether they are perfluorinated or polyfluorinated compounds. So all of these play a role and can affect the removal. In addition, we know that water contains some other solutes there, and most notably organic carbon, which is present in the form of natural organic matter, and also inorganic ions such as sulfate and nitrate, bicarbonate, and so on. So they play a role as well. And to a lesser extent, in the drinking water applications, pH and temperature can play a role as well. Just to demonstrate how, for example, water matrix play a role, I'm going to show you a graph that we have obtained on the impact of dissolved organic carbon on the performance of ion exchange resin. Here you see PFOA concentration, and on the x-axis you see PET volume treated. What it means is that the volume of the water that is treated per volume of the adsorbent, in this case resin. And the horizontal line that you see at in the middle is representing 70 nanogram per liter, which was the EPA guideline established in 2016. Now, as you can see, as the concentration of organic decreases, basically going towards right, we can see that the bed volume that allows us to reach the level by EPA, the 70 nanogram per liter, increases. So at the 20 milligram per liter, which is extremely high, we have 9,500 bed volume. We get to that level. But as we decrease, let's say 2.5, which is the far end, the blue circle, we go up to 28,000. And compare that with the DI water, we go up to 150,000 bed volume. So that demonstrates how dissolved organic carbon can play a significant role. Now, when it comes to the particular research that we are carrying out, and the target is around point of use and point of entry treatment system. It's known that these are common and they certainly can be beneficial in the interim until utilities can basically put their act together to address the PFAS at the central level, or for very small and rural communities that they don't have their utilities either the ability to treat the water, or even there is no utility involved there. So people have to take care of their water on their own. In general, POUs or point of use systems that are installed under the sink are generally more effective and more basically usable or applicable, because there is no need to basically treat to that extent the water that is used for bathing or showering. And also point of use systems are less costly, and not very many of them in terms of point of entry systems are NSF certified. Now, on the topic of certification, we know that there are multiple certification bodies. The notable one is NSF, and that's actually NSF International is the one that the most recognized one. And for point of use, point of entry systems, NSF 53 is the one that is applied. And basically 152 products or around that number from 12 manufacturers are currently certified for PFAS. And there are, of course, some that are following NSF 58, and those are more for RO systems. Now, in addition to NSF, WQA has its own certification process, which is very similar to NSF. And in fact, NSF recognizes that it just follows the same protocol for certification. And currently, there are 13 manufacturers that have their products certified. There's also sustainability certification process, which really is a third-party certification, and that is based on environmental responsible ways of producing the products. Now, these are for devices that we have in terms of certification. The materials that are used for treatment that are exposed to water, or water is exposed to them, in other words, filter media. Now, if they're used for other applications, not point of use, point of entry systems, they need to be certified, and those are NSF 61 certifications. So basically, checks or validates that when the water is exposed to these media, there are no detrimental health impacts from those media. So those are important certifications that we need to keep in mind as well. Now, how these POUs or POE systems work, basically, there are a number of studies that have been evaluated these POE systems or POU systems. Now, this is one study that has been done a number of years ago or a few years ago. And they've tested various products and monitored the number of basically PFAS. And as you can see, for many of them, we don't have particularly great removal of PFAS. And one thing I should mention is that there has been no information in terms of whether there are these products are NSF certified or not. But as you can see, many of these that are commonly used, whether pitcher products or faucet filters and so on, have not shown efficacies towards, for example, PFOA, which is the most notable one. But there are also some that are working. And these are like the far right RO systems or two-stage filters. So what really brings this question is that what POE systems or what POU systems are effective, and also what type of PFAS or how much they are effective towards different PFAS. As you can see, even for each of these POU systems, you can see that the efficacies for different PFAS changes as well. And that's what the rationale for our study has been, is that, okay, we have so many PFAS, a number of them are basically regulated, and many of them are not regulated. And in fact, studies have shown that they are going to be converted over time to the regulated one or transformed to regulated one. So what PFAS we need to think about when we are assessing or looking at the efficacy of the POU or POE systems, particularly when we are talking about absorption-based processes that are very common in point of use and point of entry systems. So that's been the rationale for our studies. And at this point, I'd like to pass on to Ehsan, who can tell you a little bit more about our study and some of the results that we have been getting. Ehsan?
Yeah, of course. Thank you, Dr. Mohseni. So yeah, as part of our methodology that I will cover in the next few slides, we selected a wide range of PFAS types, adsorptive material, experimental procedure, and water matrices to make the study as comprehensive as possible. So here, as in this slide, we are showing PFAS selection. Among 1,000 types of PFAS that have been covered in the previous slide, we selected these 16 PFAS in 5 categories of PFCAs with carboxylic functional group, PFSAs with sulfonic functional group, thermal PFAS with CH2 linkers, etheric PFAS with CO bonds in their structure, and also FASAs with sulfonomic functional group. And these selected PFAS varied in different chain lengths with 3 to 10 carbon in their chain lengths. And of course, we considered the 6 regulated PFAS by US EPA as noted by dashed lines, and also those has been considered as hazardous constituents, like the ones that are shown with the stars. So in the next slide, in terms of adsorptive material, we have tested different types of gags and ion exchange resins. In general, there are 3 types of gags based on coal, coconut shell, and wood. And we selected one from each of these categories as a representative to see how they are working. Along with granular activated carbon or GAC, we also tested 2 types of ion exchange resins that are available in the market as PFAS-specific resins. And it's important to note here is that all employed adsorbent in this study are NSF certified. In the next slide, we are showing the experimental procedures that we followed. In general, we followed these 3 experimental procedures to study the adsorption capacity, kinetic and rate of each of these adsorbents, as well as their column performance. As an isotherm study, we use a method that we call it BPI or bottle point isotherm. In this study, a mixture of all those 16 PFAS was mixed with different dosage of each of those adsorbents. And we allow it to last for 3 weeks to make sure that the full equilibrium was reached. Then, based on the concentration of the PFAS in the adsorbent phase, as well as in the solution phase at equilibrium, we are able to find out the relation between these two, such as the Fenwick isotherm study that I'm showing the equation here. In the next part, or in our second experimental procedure for kinetic study that we use a jar tester, we took sample at shorter time, like up to 60 hours, to find out how fast is the adsorption of each of these PFAS for the adsorbent. And for the third experimental procedure that we use, column study, which is the main procedure of our study, we use a well-established procedure that we call it rapid small-scale column test, or RSCT. Whereas shown here, a ground version of employed adsorbent in the next slide is used in much smaller size of the column versus pilot or real scale treatment setting. This process can predict the breakthrough bed volume of each adsorbent in much shorter time and also in lower cost. And also in the next slide, we are showing some of the important definition as bed volume, which is the volume of treated water over the volume of adsorbent. And based on that, we can define some other definitions, like for example, breakthrough bed volume, like for example, BB10, BB50, or BB100, which have a similar definition. For example, BB10 is the breakthrough bed volume, that the concentration of our PFAS in the effluent reaching 10% of the concentration in the inlet, or BB50 or BB100 have the same definitions. And in terms of challenge water and PFAS in the next slide, we use challenge water, as you can see here in the left table, with this condition with the concentration of PFAS varied in the range of 8 to 12 ppb for studying the isotherm and kinetic. And also for the RSCT or column study, we use different challenge water to study the impact of organic carbon or inorganic anions on PFAS capture with each of those adsorbents. And here, to be more close to what PFAS concentration have been detected or recorded in the natural waters, we use a concentration of PFAS in the range of 0.81 to 0.7 ppb. And moving to the result, the next few slides. So for the first challenge water that we use for isotherm and kinetic study, very briefly before jumping into the result, it should be noted that the isotherm study can show the transfer of the adsorbent or PFAS from solution phase to the adsorbent phase at equilibrium condition. So it is very important in characterizing the adsorbent capacity. And for kinetic study, it is showing the rate of retention of each of these adsorbents from solution phase to the adsorbent phase. So it's showing the rate of the retention. And it is very important for design of adsorption system, like for example, empty bed contact time or the contact time that is needed in the column system. So from the next slide, we are showing the result. So the left-hand side graphs are showing the friendly constant for different adsorbents. The left top one is showing for different GACs or GACs, and the left bottom one is showing for the ion exchange resins. The bar is showing the Friendly constant for each PFAS in each category, where blue bars are for coal-based, red one is for coconut shell-based, and green one is for wood-based. And as shown, coal and coconut shell-based GACs have greater capacity compared to wood-based GAC, particularly for short-chain PFAS. And in general, ion exchange resin have much greater capacity or with higher Friendly constant with 2 to 3 times greater Friendly constant compared to GACs. And comparing ion exchange resin between each other, ion exchange resin one is the best one for the short chain one. However, the longer chain one can be captured by the ion exchange resin too. And as shown, for obtaining each of these Friendly constant or each of these bar graphs in these two graphs, we have a graph something like that we are showing for the 4-2F case, that we are showing a Q or the amount of PFAS adsorbed on each of the adsorbent versus C or the concentration of each of those PFAS in the solution phase. And if we plot Q versus C, we can have this Friendly constant for each of those PFAS for each of those adsorbents. And as clearly showing here, ion exchange resins have much higher capacity comparing to GACs. In addition, in the next slide, we are showing the results of the Friendly constant for each of those materials that have been tested versus the chain length. And as shown here, by increasing the chain length, the Friendly constant or the adsorption capacity for each of those adsorbents is increasing by increasing the chain length. And obviously here, the Friendly constant for ion exchange resins are much greater, or the adsorption capacity for ion exchange resins are much greater comparing to GACs. In the next slide, in terms of kinetic study, that we are showing how fast each of these PFAS can be captured by each of those adsorbents. Again, we are showing the same type of result. The top left one is showing the pseudo-first order removal rate constant for GACs, and the bottom left one is showing the same removal rate constant for ion exchange resins. Again, the kinetic rate for ion exchange resins is much greater in the range of 1.5 or 2 times greater than what we have for GACs. And again, comparing the GACs together, the blue graph and also the red one for coal and coconut shell-based one are much faster comparing to wood-based GAC, particularly for the short-chain PFAS. And again, if we, for each of these graph, or each of these bar graph, we have a graph something like that for 4-2 FTS that we are showing the 4-2 FTS removal or C over C naught versus time up to 60 hours in the jar tracer setting. And again, clearly it showed that the ion exchange resins have much faster kinetic comparing to the granular activated carbon for capturing PFAS. And moving to RSCT with different challenge water. So again, we use different challenge water to show the impact of water matrices on the capacity or column performance of each of those adsorbents. And RSCT can define or determine the volume of the influent solution that has passed through the column. And when we reach BB10, BB50, or BB100, when the concentration of each of those PFAS in the effluent reach 10%, 50%, or 100% of the initial concentration in the inlet of water. So in the next slide, we are showing the breakthrough bed volume. This is a typical breakthrough curve for the GACs and ion exchange resin. Here we are showing the total PFAS concentration in the effluent of each of those adsorbent media versus the bed volume. And as shown here, in line with our previous observation for adsorption capacity and kinetic rate, GAC reach saturation at much faster bed volume, like for example below 40,000 bed volume. However, ion exchange resin were able to effectively capture PFAS at much higher bed volumes. Among GACs, coal-based GAC outperforms the other two types of GACs, where the wood-based one reaching saturation sooner, somewhere around 20,000 bed volume. And beyond that, we had some release of PFAS, which happened when captured PFAS are knocked up with water matrices with higher affinity to be captured, like for example, organic matter. And among ion exchange resins, ion exchange resin one outperform the second ion exchange resin, particularly for short-chain PFAS. In the next slide, we are showing the breakthrough bed volume of 50 for different GAC. So we obtained the same breakthrough curves that I showed in the previous slide for each individual PFAS, where results are summarized here, showing the BB50 or bed volume at which concentration of each of these PFAS reach 50% of the inlet water. So as shown here, consistently for all tested GACs, PFBA is the first PFAS reaching BB50 or 50% saturation at much shorter bed volume compared to other PFAS. Among regulated PFAS, as shown, coal-based and wood-based GAC have PFOA as the first PFAS reaching BB50, while for the case of coconut shell-based GAC, GenX reach BB50 sooner. These graphs are show the better performance of the coal-based compared to the coconut shell-based, and again compared to the wood-based GAC. And in the next slide, we are showing the same data, the same result for the case of ion exchange resin. Again, similarly, PFBA is the one that is reaching BB50 sooner. In addition, among regulated PFAS, PFOA reach BB50 sooner compared to the other regulated PFAS. It also shows the better performance of ion exchange resin 1 compared to ion exchange resin 2, particularly for short-chain PFAS. But the bar graph for BB50, for example, for the short-chain PFAS are significantly higher for ion exchange resin 1, which indicate that ion exchange resin 1 can remove shorter chain PFAS more effectively. And in the next two slides, we are showing the impacts of high DOC or dissolved organic carbon concentration for GACs and ion exchange resin. So if the first challenge water with DOC 2 mg per liter, we zoom in a part of the result, along with the result of the second challenge water, with higher DOC of 5 mg per liter, it shows that the wood-based GAC and then coconut shell-based one, to some extent, are impacted by higher DOC. If we go to the next slide. Yeah. And the next one, please. Yeah. So it shows that the challenge water 2 with a higher DOC of 5 milligram per liter. For that one, the wood-based one and coconut shell-based one are impacted to some extent with the higher DOC, where the concentration of PFAS in the effluent of these two GAC reach higher value at a lower bed volumes. This trend was more pronounced for the short-chain PFAS, where high concentration of DOC can block pores or cause some electrostatic repulsion for short-chain PFAS. And in the next slide, we are showing the same result for the case of ion exchange resin. So again, if we zoom in a part of the result of the first challenge water with DOC of 2 milligram per liter compared to the second challenge water with DOC of 5 milligram per liter, it shows that the both resin did not impacted much with the high DOC concentration, at least for the range that we are tested 2 versus 5 milligram per liter, since they are both available in the market as PFAS specific resin, and particularly it is the case for the ion exchange resin 1. And the next few slides that I will pass to Dr. Mohseni, we will cover the take-home messages and final words.
Thank you, Hassan. So just to recap what we have presented, basically. So what we are seeing from this study and also some other studies that we have been conducting is that different PFAS structures and properties lead to their different removal efficacies. The smaller chain length PFAS or shorter chain PFAS generally have lower absorption capacities, particularly for granular activated carbon. And one other thing that is important to consider, and we have not particularly shown here, is that when these short chain PFAS initially are absorbed on carbon and to some extent on resins, as the longer chain PFAS are introduced to them, these shorter chain PFAS are going to be knocked out. And if you extend the removal or treatment, you will see that these shorter chain are actually coming off from these adsorbents. So that's one important consideration when we are selecting the type of adsorbent as well. Between the shorter chain PFAS and in general, for all PFAS, carboxylic functional groups break through faster. So again, depending on what PFAS we have in our water, they are going to be coming off from these adsorbents sooner. So, and as part of this study, we are hoping to come to final conclusion and introduce the type of PFAS that could be used as surrogates, as the purpose of this study is about. Some other basically key points that we obtained is that, of course, not a surprise, but we've been able to demonstrate that ion exchange resins show greater capacity and also faster kinetics for PFAS compared to activated carbon. And then coal-based GAC or GAC outperforms other types of GACs, particularly wood-based GAC shows relatively poor removal of PFAS, particularly again shorter chain PFAS. The only exception that it did really well was PFOS, which is one of the legacy PFAS. The impact of DOC cannot be underestimated or avoided or ignored, particularly for activated carbon. In general, for the studies that we have been doing for the type of resins that we have investigated, DOC had lesser impact in there as well. So these are the key points that have been found so far. And with that, I'd like to acknowledge the team that have been involved in the work. Particularly, we had 2 undergraduate students who have helped us with this research, Tanya and Lauren. We certainly acknowledge and appreciate the support from Water Quality Research Foundation, giving us the opportunity to work on this great project. We thank the task force and the board of Water Quality Research Foundation, and Kelly and Kayla for all the support throughout. And NSERC, our funding agency, Natural Sciences and Research, Engineering Research Council of Canada as well. With that, I'll stop and happy to take any questions if there are any as well. I see a few questions in the chat as well.
Yeah, absolutely. Thanks so much, Majid. And thank you for being so efficient during this webinar. I know you answered a bunch of questions already, so appreciate that. I'll start with the top question here. I believe slide 19 and 20 demonstrated that two-stage filters were nearly as effective as RO in removal over a wide range of PFAS compounds. What type of media are these two-stage filters? I'm assuming sediment and carbon filtration.
That's a good question. Unfortunately, the information was not given in terms of the details of the media were being used in these filters. Either these could be media or the sedimentation and carbon, or another hypothesis that we have could be carbon followed by ion exchange.
Okay, next question. I am a bit curious about how you determined the isotherm model or PFO model are the most representative models, considering that there are several other isothermic and kinetic models. Thank you for your presentation.
Thank you. We've tested generally a number of them from our previous studies as well, and we found that generally Langmuir is working better. But in this case, we tested or we evaluated or considered Langmuir. I mentioned Langmuir earlier. Langmuir as well. Between Langmuir and Friendlich showed a slightly better fit to the data as well.
Okay, next question. Why does coal GAC show better performance than coconut and wood-based GAC? Does this trend depend on manufacturers?
Well, it's the detailed chemistry of that. I'm not quite familiar. I mean, someone from the manufacturing body may be able to answer this question more. But from what we know, the pore sizes and the structure of the carbon plays a significant role. But this has been, in general, what we have seen throughout. So it can't be unique to the particular carbon that we are using. Of course, we chose the one in the market that has been demonstrated to be the most effective in terms of PFAS removal. But also throughout various publications, we see other coal-based carbon performing or outperforming other carbons as well.
Okay, next question. Do you run any control testing or running challenge water without PFAS compounds?
Without PFAS compounds to remove organics or background compounds, if that's the case, no. We have done it before just to see how much removal of carbon we get from, for example, our background standard carbon is about 2 parts per million or 2 ppm. So we've done those studies earlier before, but not as part of this particular study.
And sorry for adding to that, also for the control test, we did the same challenge water with the PFAS, but without any adsorbents. For example, passing through the column, just with the, for example, the column or the glass bits for the RSSCT test to see if there is any capture of PFAS or release of PFAS as part of the control test. But as Dr. Musa mentioned, without PFAS, no, we have not done it before or as part of this study.
Okay, thank you. Is there an expectation that carbon block would perform better than a loose bed of carbon?
It's a good question. We have not done this, but when this loose bed means granular forms of carbon, If that's the case, we think that this overall surface area might be higher. So that's my guess. We haven't looked at carbon plots.
Okay, any reason for not following NSF 53 for challenge preparation and analysis?
NSF 53, to a large extent, is looking at devices as well. But in terms of in terms of challenge water, we try to use or select the challenge water based on what might represent, and in general, what the water's basically matrices are in the nation. So as I said, we started with very typical concentrations of organics around 2 ppm. So in general, drinking water supplies, if they're treated or when it comes from municipal systems, are around 2 ppm. But there are, of course, higher concentration of organics that we are using as well. So basically, we are looking at the ranges of both organics and inorganics in the water.
Okay, next question. What kind of DOC source did you use?
We generally use more of a humic nature, which are generally larger molecular weights, and they're known to block or occupy the pores of the adsorbents. So it is, I mean, if they are of a lower molecular weight nature, they probably have lesser impact, but we want it to be a little bit more on the so like the upper side of the blockage of the pores. So that's why we use of a humic nature.
Okay, next question. What is the chemical compound used to adjust DOC?
It's the same answer, the same as the previous one, of humic nature of organic matter.
Is the contact surface of active carbon of importance, GAC, compared to powdered carbon?
Well, compared to resin and carbon, definitely is important. So in general, it all depends on the kinetics. So the contact that we have chosen in our study are based on the kinetics that we found. So carbon, as Ehsan showed, has lower kinetics compared to resin, so we allowed for larger contact. Now, compared to powdered activated carbon, powdered activated carbon generally is not used in the POU or POE system, so we haven't really tested these ones. But in terms of applications of powdered activated carbon, in municipal systems, they are generally used for basically temporary measures to be able to basically temporarily address the PFAS issues. They're introduced in the water and they need to be separated. So the sludge production is another issue. So kinetics is expected to be a slightly better because higher surface area, but for the context of the work that we have been doing, which is point of use or point of entry applications, no, we haven't used powdered activated carbon.
Okay, last question of the day, and I probably will not phrase this correctly, is coming up with a single surrogate parameter for PFAS analysis the objective of this research? Something like a COD or TOC in the case of general organic contamination. Further, is there such a parameter where PFAS is considered or are considered?
Not really. We are not planning to use that as a measure. If you want to think about it, we can think about total organic fluorine as being a single parameter. Yes. So from that perspective, one could come up with one parameter that you can say to be monitored. But that's a very difficult one to do. So not very many labs are equipped as well. And also, it's going to be a little bit overkill from a regulatory point of view, because in my view, if a supplier is supposed to remove all the fluorine in the water, depending on the source, it might be challenging. Again, I'm not in a position to suggest what the manufacturers are going to basically produce and so on. But I would say even for them to address the current regulatory basically framework that is existing, it's going to be a challenge. So going back to the surrogate, what we hope to achieve is to suggest one or a few specific PFAS that could be used as a surrogate in terms of looking at or considering the efficacy of the system in terms of removing the remaining regulated and the remaining PFAS that might be of a health concern. And those are definitely more than 6. So currently, based on the EPA method, there are 29 or even a little bit higher number of PFAS that are of concern that can be detected. So should a utility or should a manufacturer go after all of those to demonstrate the efficacy of their products, it's going to be a challenge both from an analytical point of view, but also cost point of view as well. So we hope to find 1 or 2 or a few that could be used. Yes. Hope that answers the question.
Well, thank you so much to both of our speakers and the researchers today, Dr. Moshemi and Dr. Isfahani. Thank you so much for joining us and sharing a little bit about the research. We look forward to the final report coming out soon. And for everyone who's on the call, we do have our final summer school webinar planned for Tuesday, August 27th at 11 a.m. Central, 12 p.m. Eastern. Please plan to join us for our final summer school session. We'll be talking about WQRF's 2025 call for poster presentations, and we'll also share 2 presentations from 2024's call for posters, our 2 winners, one of which is actually from University of British Columbia, as well as Virginia Tech. So we look forward to hosting our poster winners on August 27th. And for everyone who's on this call today, thank you so much for joining us. We look forward to seeing you then. Have a great rest of your day.
Thank you.